Enhancement of COPD Biological Networks using a web-based collaboration interface. So biological network models depict the interrelationships between multiple signaling pathways and how their perturbations may dysregulate biological processes, eventually leading to the disease. The generation of this set of networks relied on two approaches. One is a manual curation of published literature, and the second is a data-driven reverse causal reasoning methodology. So this network pertains to COPD, a chronic obstructive pulmonary disease. So COPD is a progressive inflammatory disease that is induced by cigarette smoking, among other things, as well as inhalation of pollutants, dust, chemicals, and other foreign matter. And this ultimately manifests as tissue destruction in the alveolar compartments and also airflow limitation. And this leads to reduced oxygen exchange. So COPD affects a wide spectrum of biological processes in lung tissue, such as oxidative stress, inflammation, apoptosis, proliferation, and senescence. So as recently reported by the Global Initiative for Chronic Obstructive Lung Disease, current pharmacologic therapies cannot cure the disease, but only reduce the symptoms and the frequency and severity of the exacerbations. So it appears most efficient to target COPD at the earliest distinguishable state when the extent of the irreversible damage is still small. COPD network models are important tools to better understand the biological components and processes underlying initial disease development. Since smoking cessation appears to be the most efficient therapy in smoking-related COPD, the models of early onset COPD can also be expected to be valuable tools for the development and testing of reduced risk products that may prevent COPD progression in a comparable manner as cessation does. BEL, or biological expression language, was used to represent the biological relationships within the network. So Bell language is a proprietary language developed by Solventa, and it is based on literature. So the highly specific language is computable, and each edge that the cell represents is based on a literature reference. In this example, transcriptional activity of IRF5, which increases the protein abundance of IL-6. That's the name of the edge. The edge is from the literature reference, and the paper specifically stated that virus infection of human monocyte cells induces IRF5 transcriptional activity, and this increases the expression of IL-6. So what the Bell language is able to capture is this computable edge where there are different prefixes for the very specific activity that is being represented. So IRF5 is the transcriptional activity, and this is represented by T script of the protein IRF5. And HGNC represents the namespace that IRF5 is in, the Human Genome Nomenclature Committee. So we have different examples for namespaces for protein complexes and biological processes. So we have this increased arrow that you will see in the network, and then leading to the subject of the relationship, the protein of IL-6, which is also in the HGNC namespace. We also have a context that is annotated with each edge. So we know that this comes from human monocyte cells, and these can be filtered as desired using the Bell language. So all of the edges that you will see in the network are supported by the peer-reviewed scientific findings. The construction of the networks can be divided into three phases. In phase 1, the networks reported here were created from a corpus or body of literature that would be appropriate for this particular model. There was a focus on lung biology, especially COPD. Other diseases such as asthma were not investigated. So once this body of literature was selected and a model was built, the model was used for the addition of edges and nodes from the knowledge base that Solventa has. In addition, some of the data-driven nodes were also added, derived from transcriptional profiling data that are publicly available. This transcriptional profiling data was used to apply reverse causal reasoning, that is an algorithm developed by Solventa to derive upstream controllers. Those upstream controllers and nodes were also added to this model and were used to connect edges derived from the knowledge base. In phase 2, these networks were made available online to the entire scientific community for critical review or crowd-based curation during the Network Verification Challenge, which is an open phase under the umbrella of the SBB Improver project. In phase 3, finally, a prioritized subset of these networks were discussed during an in-person Jamboree meeting, where the crowd-submitted revisions were reviewed and decisions to improve the networks were finalized. So the final versions of the network are available at the SBB Improver website. In the next few slides, I will go over each of these phases in detail. In phase 1, initially, there is the process of literature curation. A broad survey of literature was conducted to locate the studies that investigated the mechanistic biology of COPD pathogenesis. So potential COPD biomarkers were collected from sputum, from alveolar lavage, and mouse and human blood samples. And mechanisms that regulate COPD processes were gathered from the literature and curated. 90 non-diseased lung networks previously published in the areas of cell proliferation, cell stress, inflammation, DNA damage, cell death, tissue repair, and angiogenesis were used as the initial scaffolds for the COPD enhancement during phase 1. A set of 90 networks were agglomerated by the expert team to yield a more concise set of 50 networks. And these 50 networks combined and standardized complementary cellular pathways. And you can see these networks here, which are based on cell stress, cell fate, cell proliferation, etc. So as an example of this consolidation is the merging of the three non-disease networks related to T helper 1 cells, that is Th1 differentiation, Th1 response, and T cell recruitment and activation. And all of these were consolidated into a single new Th1 signaling network. So here is the list of the original models that correspond to the agglomerated models, along with the description and a link to the URL of the networks in the NBC website. Next, RCR analysis was performed on human COPD gene expression datasets in the public domain in order to predict potential mechanisms. These were gene expression omnibus or GEO COPD and emphysema datasets from lung, small airway, and alveolar macrophages of early COPD patients and healthy smokers. So these are available for the public to view. So RCR had been used previously to predict upstream regulators from transcriptomic data. Mechanisms that were predicted by RCR to be active and that were not already incorporated in the non-diseased networks were vetted on an individual basis to locate supporting literature for their potential involvement in COPD pathogenesis. As such, this important aspect of network construction potentially captures those biological components that may have emerging roles in disease progression. Networks that were not enhanced with COPD-specific mechanisms from the literature, or RCR, included the DNA damage and Notch signaling networks. Although both these networks were relevant to the development of COPD, they were not augmented beyond the original non-diseased network scaffolds because no studies on the differences in signaling between non-diseased and signaling diseased states were available. So for a detailed explanation of the concept behind reverse causal reasoning, please visit the webinar section at the SBV Improver website. During phase 2, a global community of scientists participated in the network verification challenge by contributing the expertise to one or several of the network models. So scientists could contribute by verifying existing evidence for network edges using a system that allowed users to vote on evidence to indicate agreement or disagreement with its appropriateness within the network structure and boundary conditions. So in total, 50 network models received 2,456 evidence votes, 1,795 of which supported the confirmation of evidence and 661 favored the rejection of evidence. So here is a homepage of the network verification website. Let me navigate to one example network, for example, the macrophage signaling network. So here is the macrophage signaling network, and you can see that it's quite a large subnetwork. And so the basic representation you see here is that you can see that the nodes are represented using different shapes. The protein abundance terms are represented as this pale blue square. The kinase activity terms are represented as purple triangles. So in terms of navigating the networks and finding specific nodes and how they are encoded in the Bell language, you can access the node list on the top portion of the network viewer panel. And this gives a total of 232 nodes found in the macrophage signaling network. And this is a way to browse how different biological terms are encoded in the network. And you can zoom in on specific terms that you may be interested in. So here, if you select the kinase activity of, for example, the SRC family, you can see that we are taken to a zoomed in representation with the kinase activity of the SRC family sort of centered in the middle here. And this is connected to multiple different entities in the network. So on the edge panel in the right portion, it indicates a list of all the edges that are found connected to the SRC family term. And this is also an easy way to navigate through and find specific relationships in the networks that you are interested in. We can also filter the nodes based on function types. You could easily, for example, select the kinase activity. And this gives one method to filter through the specific function types of the network. So we see a collection of all those kinase terms. You could also filter this by biological process. And this gives a list of 11 Bell terms that are associated with the biological process found in this network. From this, we can select a biological process of interest to us. Let's take, for example, phagocytosis. So this allows us to look at what entities in the network may influence phagocytosis. And in this, we see there are a total of 11 edges. One example would be that lipoxin A4 increases the process of phagocytosis. So we select this example. We see two evidence lines from human macrophages that support this particular edge in the network. And if you were to click on a specific evidence line to investigate the literature supporting this particular edge, in this case it's human macrophages. It's a publication from the Journal of Immunology in 2000. And it indicates a relationship between lipoxin A4 and phagocytosis. And then you can approve or reject the statement as you see fit. So there's also an option of creating new evidence that supports this edge. So this would be the dialog box. And you can indicate the specific PubMed ID and the contextual information that the publication would be supporting. So we can also add new edges. So let's say in this case I selected ARG1 and the only edge that currently exists in the network is this connection as a biomarker for macrophage activation. And let's say we found another example that relates ARG1 to a different entity in the network. So what we can do is use this panel to create an entirely new edge. And so if you were to click on this, you would go to this page. And this is a new edge form where you can input the relationship between the subject and the object. And finally, you can add evidence to the edge and also verify your work. So in phase three, to represent the crowd community, the top 20 active performers who created the most pieces of evidence and submitted at least 20 votes during the network verification challenge were invited to an in-person jamboree to discuss the network refinements as a group. Additional subject matter experts in the fields of COPD, lung biology, and biological processes, basically represented by the networks, were invited to participate in the discussions and contribute their expert feedback independent from the network building experts. So during the challenge, participants worked individually on the website, adding individual edges, but they did not have the capacity or the ability to make major changes to the structure of the network models. So the in-person Jamboree discussions was an opportunity to implement larger or much broader changes to better represent the biological processes as they related to COPD. So 15 of the networks were prioritized and selected for discussion during phase 3 based on the level of crowdsourced activity and the importance in COPD onset as considered by the network building experts. And this phase 3 provided an additional layer of verification for the online enhancements. So, three of the network models that had received the largest amounts of crowd activity, namely neutrophil signaling, macrophage signaling, and Th1 signaling, also underwent significant additional enhancements to improve the granularity with respect to COPD onset and pathogenesis. So these enhancements were incorporated to improve the connectivity within the network structures. So in one example, the apoptosis network model, the original network pathway indicated that the X-ray repair complementing defective repair in Chinese hamster cell 6 or XRCC6 protein decreased the process of apoptosis. During the phase 3 discussions, additional literature evidence provided a more detailed mechanistic understanding of this phenomenon. XRCC6 was reported to decrease the activity of the BCL2 associated protein, which is known to increase mitochondrial permeability and therefore promote apoptosis. The overall effect of the negative regulation of BAX by XRCC6 was therefore a decrease in apoptotic cell death. By improving the granularity of this pathway in the apoptosis network, a more comprehensive representation was achieved for components that are related to critical cellular processes mediating disease onset. A similar improvement was incorporated into the mechanisms of cellular senescence. This network model, the original network pathway, indicated that the chemical acreolin, which is a common component of cigarette smoke, increased cell senescence. And you can see this in figure B. So during phase 3, the pathway connecting these two components was expanded using additional literature evidence. In several studies, acreolin was found to decrease the activity of sirtuin-1, which is a known negative regulator of the FOXO3 transcription factor, and the FOXO3 activity is known to promote cellular senescence. Therefore, the overall observed effect was acreolin acting to potentiate cellular senescence in exposed cells, which is a well-characterized mechanism of action for this toxic chemical. Again, the generation of more comprehensive network models of biological process in close proximity to disease onset allowed for a greater mechanistic understanding of how environmental factors can contribute to COPD development. So here is a summary of the number of nodes and edges for each of the different phases, with a breakdown of the networks that received the largest percent of the enhancements. So here you can see macrophage, neutrophil, and Th1, Th2 signaling. And you can see, for example, that phase 1 received 200 additional nodes and 487 new edges, and so on. So with this, I end my presentation, and I would like to thank you for your attention.